Hundreds of thousands of researchers around the world are working to improve life and address imminent threats to humanity. Often their research ends up in the scientific valley of death in the form of publications and patents that never see the light of day. That is about to change. Welcome to the Lab to Startup podcast, hosted by Naresh Sunkara, founder and executive director of the Berkeley Postdoc Entrepreneurship Program at the University of California, Berkeley. This show has two main goals: share the stories of those who have successfully founded startups based on their own research, and highlight resources needed to help those aspiring to launch startups in the deep tech space. Whether it's electric cars, vaccines, addressing climate change concerns, or possibly establishing life on other planets, Naresh and his esteemed guests want to help scientists, engineers, faculty, and researchers bring their innovations to market. Learn more and subscribe today at labtostartup.com. And now, here's Naresh.
My guests today are Karthik Balakrishnan and Anand Kesavaraju, the co-founders of Nodexus, a biotechnology startup that is developing scientific tools to accelerate biological workflows and make them universally accessible. The first product is a single-cell sorting and dispensing machine. In this episode of Lab to Startup, we talk about the problems around single-cell sorting and the solution they came up with, the evolution of the technology, challenges and advantages of growing a lean operation, identifying markets, mentors, and investors, and some of the lessons learned building a scientific tools company. Karthik and Anand have been very good friends of mine for a long time. It's always fun to bring on close friends where you can dig deeper into some of the topics. And I really hope you enjoyed this conversation. Anand, Karthik, welcome to the show. So glad to have you guys here. I think the best place to start your story is going to be talk about the problem you're solving at Nodexus and how you came across the problem, and we go into the solution afterwards.
Definitely. Thanks, Naresh, for having us. Looking forward to the discussion.
Yeah, we've obviously known you for many years, and you've seen us through different stages of the company evolution and the product evolution, but great to be here.
Awesome. Let's get into the problem. State the problem you're trying to solve here.
Yeah, certainly. For our flagship NX1 platform, and really taking a step back, actually, one of the goals of the company is really to commercialize tools that make biological workflows more efficient and more accessible overall. And in our initial product, which is the NX1, we're doing this by leveraging microfluidic technology to make single-cell sorting and isolation universally adoptable. Cell sorting and isolation are possible today, but there's a number of challenges that make them a little bit difficult to bring into every lab and every facility. And that's kind of what we're trying to do with this platform.
What is hard about sorting?
That's a very good question, and I'm glad you asked. There's quite a few things that are very hard about sorting. The first thing is the biological question, which is, am I sorting what I should be sorting? The separate piece of that is very foundational to the way that we think about building technologies, and it's really around ease of use and affordability. Like Karthik mentioned, there are tools out there. They tend to be multi-100,000 all the way up to multi-million-dollar types of systems that typically are only found in dedicated centralized facilities with expert PhD or MD level training behind these folks that know how to operate these machines and tend to be the only folks that are allowed to operate these machines. And so when you think about those two pieces of it together, there's kind of this dark art around cell sorting, which, hey, it's restricted very much so to only the people that know how to use and can delicately tune these very expensive systems. And then there's the deeper biological questions of, hey, what is actually happening to my cells during this process? Why is it that sometimes I get plates that come out and have cells that are great? And why is it that many times they come out of the cell sorter at the centralized facility and they're all dead? Or I've spent weeks or months preparing for something that ultimately doesn't work. How do you try to provide solutions that lead people to be able to solve those problems in their own facilities, in their own labs?
What you're saying is it's very esoteric. Not many people can access the technology and very expensive. And there's a biological question of not being able to tell what you're sorting there. Let's go a step deeper into that technical aspect of it. What are the technical problems that exist with the existing technologies? And how much better are you guys? And what is that technical innovation you came up with that makes you better?
If you look at the current technologies, or the conventional technologies, I should say, there's a number of aspects of the approach that's currently widely used. If you're thinking about FACS, centralized facility-based sorting, And that can fall into a number of buckets in terms of how you think about the value proposition we are offering based on the technology that we've kind of developed within our company. And conventional FACS is extremely complex to operate. There's a lot of setup and tuning that goes into the process. That's one of the major reasons that these systems are typically restricted to core facilities managed by very, very skilled experts in terms of setting up, tuning, running the machines. Typical use cases, I may be in an academic lab or a small company, and when I want to sort some cells, my workflow involves booking time 3-4 weeks in advance at a core facility. If I'm in the Bay Area, that could be a Berkeley core, that could be a Stanford core, just as examples. And I have to have that expert technician or core manager take my samples, run them on the machine, which involves setting up the machine, getting it operated and optimized for my run, giving me back my cells and returning to my facility. So just by nature of the way the existing kind of conventional FACS machines operate, there's a ton of tuning, calibration, drop delay, the details in terms of the operation that are very, very difficult to pick up by just any novice user. So what we're trying to do is demystify that process, make it easily adoptable, easily usable for anybody, not just a flow cytometry or FACS expert. The other piece of it is the fundamental methodologies that are used in these FACS systems. They can potentially cause damage to the cells that they're analyzing, they're sorting, just by the methodology that's being enacted through these platforms, where they're running cells at a high pressure through a flow cell, deflecting droplets to have them land in targeted areas within a microtiter well plate.
With some very high voltage involved also.
So it's the high pressure and the voltage that's killing the cells.
There's many hypotheses as to why these things happen, but certainly when you think about fragile cell populations and really the whole field of biology, or multiple fields of biology, I should say, are trending towards these very fragile or cells that don't like to be perturbed too much. So even taking them out of their cell culture media, resuspending them in a harsh buffer or sheath fluid like you will have to do with traditional pieces of instrumentation, that in and of itself can be dangerous to cells. And then once you then apply high pressure and high voltage to make sure that the cells actually end up in well plates or collection tubes, what have you, that process in and of itself, there's been many studies out there that have shown that that can be very damaging to intracellular processes and just generally the cell health overall.
Thank you for framing that problem there. I think that's very, very helpful.
I was just going to add another pain point that we encountered during our interaction with these customer segments, and that's related to the contamination aspect of running through a centralized facility piece of equipment, where you have to go through extensive cleaning and bleaching procedures in between runs. And even then, there's always some concern of, was there any remnant sample? Can I be 100% confident that the sample I'm running isn't going to be contaminated? And these are the types of functional and operational pain points that we set out to address with our platform.
So let's go into the solution. So what did you guys come up with, and how did you solve those problems there? If you can talk one problem at a time.
At a high level, our NX1 platform, the flagship that you might find on our website or in some of our materials, really intended to be this extremely easy to use, should be the most affordable and most accessible cell sorting solution out there, and intended to be something that sits on a benchtop or in a tissue culture hood without needing dedicated facilities, dedicated space, which we know is a big problem for companies that are trying to grow, or even academic labs. And even the biggest core labs and the biggest companies, biotech, pharma, and so forth, where space can be a huge premium alongside operator ability or researcher time and training time and various aspects like that. So really the heart of the system is around microfluidic cartridges that are part of our company's foundational history. And a lot of the expertise that we have in-house is really around these disposable consumables, which partially solve the contamination concern that Karthik was just highlighting a few moments ago, but also offer a very one-click workflow for an end customer. So the typical workflow that we described, including some of the alignment and tuning and drop delay and all the extensive prep that goes into running a high-end FACS or flow cytometry system, we've really taken all of that out of the user's hands. The idea is that a user loads a cell suspension into a microfluidic cartridge, they place it into the instrument, and more or less after they load their collection plate or collection tubes, that ends their interaction with the hardware. Everything else is then done in the software where they will gate their populations or subselect their populations like they would traditionally. It's a very seamless, intuitive interface meant to be very easy for people to follow along with. So there's not a lot of knobs or buttons or things that can be changed. That's been some of the fundamental design that our team has put into the system from both the foundational hardware as well as the software development levels. And really what we say is within a matter of minutes, any user, even folks that have never touched a flow cytometer or a cell sorter, should be able to be up and running with the system. And that's not something that can really be said for any other system on the market.
As a first-hand user of fax and cell sorting, I know the pains, and I wish I had something like this about 10 years ago when I was a postdoc. Thank you for walking us through the solution. Let's stick to that thread. Can you talk about the ideation process? Where do you come across the problem? Maybe you can stitch into your backgrounds and how you came up with the solution.
Yeah,
and how the solution evolved. How about that?
When we founded the company and spun out of UC Berkeley, there was technology that we were leveraging that was really capitalizing on microfluidics, but more for cell screening and some aspects of cell sorting. But really what happened after that is we started with very cool technology, went out and did really our first layer of proper market research after founding the company. So kind of a little bit of a non-traditional process there, and really got a lot of interest in these applications that were growing very quickly. Obviously, spinning out of Berkeley, CRISPR, there was a ton of talk and excitement about the tools and how that would transform the types of work that would happen in gene editing workflows. And just using that as an example, we basically had a lot of interest in terms of applications where you can quickly process a sample, sort out your transfected cells, isolate them individually, do cell line development. So that's one example of where the motivation for making a very efficient, easy to use, affordable single cell isolation platform came from. And once we had that feedback, the initial steps were, okay, it was myself, it was Anand, and then also our VP of engineering that really helped take the lead in terms of how do we develop this from scratch? Because you have to remember, we didn't necessarily have the technology to accomplish what the market was telling us was needed. We had some foundational technology that could do a lot of cool things, but we weren't necessarily convinced that, hey, should we build the company longer term around what we started with? And once we got that feedback, it was really our VP of engineering that took the next leap in terms of foundational R&D, really starting from scratch. How do we develop out on-chip sorting that can accomplish these pain points that we identified? Contamination-free operation, affordable in terms of instrument and also consumables, and also high viability enrichment and sorting where you don't apply high pressures to these cells. So use microfluidic technology, make it very, very low pressure, and don't damage the cells during the processing at all. So we started really from ground zero after spinning out the company, which is kind of an interesting path to take. But our VP of engineering certainly took the lead, and he had prior experience commercializing products from inception to scaling at Fluidigm previously. And that was very, very worthwhile experience for us to gain insights from.
As much as you think it's non-traditional way of spinning the company out and then finding out product market fit, I think that's a common theme I'm finding with a lot of startups. It's hard. Finding a product market fit is really hard. I think this is just for the note for the audience as well. Let's go back to the topic of market research. If you can walk us through that customer discovery process, I'm sure you hear a lot of noise and then there are signals. How do you sort out this signal from the noise, and walk us through the decision-making process?
Karthik described some of the initial market research that we've done, but as you know, and I'm sure many people in the audience know, these aren't static processes or one-time things. This is something that needs to happen constantly to make sure that the biggest failing you could have in a space like ours or just broadly across entrepreneurship is you think that you have the right answer and you're going toward it, and then you stop listening to the market. And then either something passes you by or you're not keeping track of your competitors or what else is out there, or just broad changes in the market that are telling you, hey, you might make a product that hit every one of the requirements that you thought about 6 months ago or a year ago, but it turns out people may not care about that today. So we go through these cycles periodically to make sure that we're continuing to iterate. And it's caused us to pivot numerous times throughout the years that we've been in development. But I think you had asked a little bit about the logistics around customer discovery more broadly. So a lot of what we did was, at this point, it's kind of classical teachings between the lean launch pads and the NIH I-Core type of programs, the latter of which we were very fortunate to have gone through some of our NIH grant funding in the past. But really, it's trying to just get conversations with end users all the way up to folks that may be purchasing your system, or maybe decision makers, or maybe just partners along the way. So trying to make sure that the solution that's being offered actually ends up meeting the needs for every piece of the value process. So making sure that every stakeholder that you interact with either sees value in it directly or can see value in it for their organization or their facility or whatever it happens to be, and making sure that you're asking the right questions. So it's not just oh, hey, sure, I'll say something that will make you happy. That's the opposite of what you want. You want to make sure that people are being very blunt and honest with you that, hey, this is not something that I would ever use. And as you mentioned, that can either be noise or that can be signal. So you have to figure out, based on the data and based on the amount of positive or negative feedback that you get, that maybe this is something we should pursue, or maybe this definitely is not something we should be thinking about at this time. Or if we have some hubris about it, it's okay, maybe the market's not ready for it yet. Maybe we shelve it and try to bring it back in the future if that is something that we think is cool or compelling.
Was it a democratic process where the yeses were more than the nos? Or was there a gut feeling involved where even though the response towards no was more, you decided to take that leap of faith? What was that deciding factor if you had that one moment where you said like, okay, this is what we're going to build?
I think for us, there were certainly many discussions toward coming to these decisions, but we initially had focused on applications, one being circulating tumor cells, where at the time when we spun out the company, it was a very, very hot space. There were easily 30, 40 companies, and not even naming the ones that maybe are behind the scenes doing stuff that we wouldn't know about. But it was a very exciting space. And we were really at the point where we could leverage the current technology and try to see what was possible in that application space. But there were still some question marks in terms of clinical actionability, clinical utility. And it became a decision internally in the company of, do we go down that route and build this out really from a personnel standpoint, a mentor and advisor standpoint, where we get clinical leaders to really help drive the future of the organization. But we also wanted to continue our market research, because one of the tough things I think we had come across was when you have a versatile platform technology, I think that's a blessing and a curse, because you think you can solve everything, but you really do need to kind of focus and see, where should we refine this? If we're going to put all the effort in terms of R&D, how do we point this and position this effectively? And for us, we started getting a lot of feedback on single cell isolation, on even applications that were preclinical or in the RUO space. CRISPR, I mentioned, being one of those technologies where we could try to piggyback on workflows. So if you looked at even the types of number of publications going on, industry leaders talking about these technologies, for us, it was I think through our customer discovery interviews, very, very compelling conversations and excitement of, oh, I would love to use this now, and I think I would use it even more in the future, versus conversations saying, oh, this sounds like a cool kind of technology. I could see myself once in a while using it, or maybe if I could figure out this one application, it could be useful. Sifting through and seeing where there was value beyond it being kind of a cool toy that may be a paperweight on a lab bench, to something that was more, I would love to use this now, that could boost my efficiency immediately. And you have stakeholders at all points in the organization vouching for that. It's the user, it's the person that can make the purchasing decision. It's really everybody involved that can make it a compelling business argument, in addition to a compelling science argument, I think.
Interesting. These are all tough decisions. The reason I'm trying to engage you in this conversation is try to get the insights. And speaking of decision making, you both were the co-founders. Talk about the co-founder chemistry. How has it evolved? You've been together from 2014, or I'm sure earlier. Talk about how that relation evolved, about the differences and how you sorted those out and how we got till here. There must be some fun stories. I'm looking for the fun stories.
Sure. I mean, you can start. We have known each other for quite a while. Like we mentioned, we spun out of a lab out of UC Berkeley, and we had actually, Karthik and I had met in that lab many years ago. Let's put it that way, to be diplomatic. I would say we've been colleagues, we've been friends, we've been roommates. Chemistry-wise, I don't know what to say. Sometimes I put up with him. Maybe that's no.
I think it's a very fortunate thing to be able to fortunate, maybe almost a requirement of like when you're going through this long of a process, there's so many challenges, ups and downs. Who are people that are going to be someone you see yourself working with and getting through the challenges with? Because I think when everything's going perfectly well, it's easy to get along. It's really when the things are going south, you're going through big challenges. Is this someone I can get through that with? That's what I think any founder should ask themselves when you're thinking about founding team, or even team beyond the founding, those early folks within the organization that you're working with. And I think there's a comfort in the relationship where you don't want an echo chamber, but we want challenging ideas, or challenged ideas, I should say, where we shouldn't both be saying the exact same thing and it's always right. You should be comfortable to the point where you can disagree. And I think we've somehow managed to do that in some degree. I would say that's the same thing with our VP of engineering, who's been with us for a long time now, where we can comfortably disagree with each other, but also come to an agreement and really a cohesive path in terms of what decisions and direction the company should be taking. And I think I never take it for granted the fact that if I didn't have the right people that I was working with through some of the lows that we've gone through in the challenging times within the company, I don't think there's any way we'd have made it out the other end. It's been a very fortunate mix, especially when you have people that are willing to take on every aspect of work. It's not just, oh, I'm going to stick to my siloed role. I think that's very, very important. And I think I would say that's definitely true with Hanny, our VP of engineering, but also Anand. And I think that's one of the reasons we've been able to put up with each other for this long.
Well, Anand, a low point, maybe both can take one: pre-formation of the company, one post, and how you survived. Because I don't get to ask too much about the chemistry of co-founders. I know you guys really well, so I thought I'll take the freedom. If you can, whatever you can share.
Sure. Pre and post founding. Okay, let's see.
Where you like, I cannot work with this guy was the decision, but he came out of that.
Does yesterday no, I'm kidding. I don't know about pre. That's a hard one, actually. So let me do something in between.
Let's go with the post.
Yeah. So post, I would say it's a hard road. I mean, you know this. I think we all know this at this point, but this can be a very lonely, a very hard road, especially if everything else in your life can be going perfectly. And if the work is not going well, that's obviously one layer. But when things are hard in every respect, startups are not the most stable. They're not the most forgiving, and there's not a lot of redundancy, right? That's not the way that this works. The way that it works is you kind of give it everything you got, and then you find more and keep giving it that, and then go back home and deal with whatever you need to afterwards. And honestly, I think a big piece of it for me, and hopefully for a lot of other people that may be listening, is you have to find people that you can laugh with. You could be in the lowest position. You could be so frustrated that, oh man, I walked out of that investor meeting and we had such high hopes and we were banking on this, and they basically laughed in our faces. That could have happened. Or maybe they're more polite than that, but for all intents and purposes. But as long as you can walk outside, or maybe you cool down a little bit, but afterwards, at least you can take your mind out. You can laugh. You can have something that is not part of having someone that you have that comfort and ease around, I think is such an important part of this co-founder journey.
Absolutely.
You can find someone that might be the perfect fit for you on paper, but if you're not interacting with them in an outside of the office or outside of work capacity, this can be such a lonely path for anyone.
Indeed. I think being able to laugh is such an important thing. While you're on the subject, talk us through your evolution of the team. How did the team grow over the years, and how you were able to attract some good talent? I see a lot of good people on your team. So walk us through that process.
We've had a long, hard road where for a lot of the time, earlier days in the company, it was myself, Anand and Hanny, really getting through. It's not a unique story. Long hours, trying to take care of, really handle everything that's going on. And I think going through that for years has given us an appreciation of when you can get people to come in and help you out. And that's something we have been fortunate to be able to do, hire people in all aspects of the business: engineering, manufacturing, biology and applications, HR, really every facet of the business. I think we are at a position now where we look around at the team and we feel lucky to be working with people like this. What I will say is, even before we were able to get to a comfortable position to expand in the way that we kind of wanted, we were always surrounded by, I think, a number of very, very good, helpful, friendly, but also very critical and instructive mentors and advisors, and also investors, some folks would straddle kind of all of those roles. But I think those resources and some of the folks that may not have been employees, but still guided us, I think that's one of the reasons we were able to stick through the trenches with just a few people to get to the point where we could expand the team.
And what are the lessons you learned from the hiring process?
Oh man, it always takes so much longer than you think it's going to take. I think every startup founder is incredibly optimistic that, oh man, when someone hears my vision, they'll just jump in. They'll give their 2 weeks notice and be here and be pulling the same hours, be doing everything. And not to say that our team isn't. And I think we've been very careful and very selective in who we have brought on to the company. And I hope that we continue to do so as we scale and grow. But it's a very different mentality to think about someone who works or who has only worked, for example, at gigantic companies that may have
all the resources and the money and the time.
Exactly, all the resources. And again, it's not like those folks don't have unique challenges that they have to go through. There's a certain amount of, not even a certain amount, there's a fantastic amount of freedom that a startup can provide. There's a lot of, you can be unshackled from bureaucracy or red tape or all these things that people like. But I think it's been very important to try to find individuals that will thrive in the environment. We certainly have structure, and we've learned to have structure over the years, because when you don't have structure, things can be so free flowing that it becomes very hard to obviously rein things in, make sure that everything is focused and aligned towards the process that you're trying to work on. But having individuals that are excited about building things from the ground up, I think that's been a key thing that we've tried to look for. And something that we ask in our interviews all the time is, what is this thing that you've built? What is something new that you've introduced? Because if they can speak about that passionately and it comes through very evidently in the way that they communicate, I think those are the types of people that are very good fits for startups, even if they've never worked at a startup before, just because that process of, hey, I found out what was wrong means I need to have been able to identify that, meaning I'm observant, I'm curious. I came up with a solution means that I, again, found things that either were quote unquote off the shelf or I did it myself, which we're a very big DIY team. We've been that way by circumstance for a long time, but now we very much so try to prioritize doing that just because the amount of control and knowledge that it gives you, as opposed to just relying on external forces all the time. And then folks that can convince others and bring others along with them. I think that's very important when you're building teams. And we want people to have ownership of their work. But as Karthik mentioned earlier, we don't want people that are siloed. That can be such a detriment in a small team, as folks that think, oh, all I work on is this, and I'm not going to get involved. I don't want to ask about something else. Even if I might know a better way, I probably shouldn't do that. We can't afford to have people like that at the stages of companies that we have.
Passion about building things is something you cannot fake, I think. That's a good point to look for, at least at a startup. Speaking of building the teams and the company so far, the obvious question comes up is the money. I've seen on your Crunchbase profile that you raised about $30 million. Walk us through that process. Was there friends and family involved? And these are the stories that are told in Valley. And what have you learned about the investors? You must have interacted with a lot of angels, VCs, and what are the lessons learned in that process of getting this far?
It's certainly been a long road, and we really dipped into many of those categories that you described. So we had a few SBIR grants that were definitely very, very helpful, leveraging our technology and our platform in a number of different areas. We were very, very fortunate early on to have some very supportive angels, but also a handful of smaller venture capital firms that really supported us through the early days and our early growth initially. And those folks were very, very instrumental. They were buying into something at a very, very early stage. You had that mix of angels that are very keen and interested on the application side of things. You have some of the smaller VCs that are really taking that bite early, but they see a big market opportunity. Along the lines of beyond just capital, we also went through Skydeck, the Berkeley Accelerator, StartX, one of the Stanford affiliated programs. So resources like that, resources like QB3, very, very helpful. We mentioned the NIH I-Core that was affiliated with one of the grants that we were fortunate to receive. So really trying to get help in any way possible. And some forms that was capital, some forms that was advising and network and things like that. And really we've gone through the journey of from those initial angels that were buying into primarily a lot of the team, but also some of the applications, and they may have not been as in the weeds on the technology side, to now with our last round of funding, getting very, very sophisticated investors that are familiar with the market, the opportunity here, but also the technology and this level of doing competitive analysis. What else is out there? They've had successes with other products and companies in the broader space. So I think we've seen that full trajectory. And I think we've learned a lot of lessons through the course of getting to even where we are today. And one of those I would say is there's no one size fits all. I think any investor, whether you're talking about one angel versus another, one smaller or micro VC versus another, or even a larger firm, really depends on what firm you're talking to, what folks within the firm you're talking to. And I think one thing I would say is we are very, very lucky such that almost every one of the investors, if not all of them, have been very, very good to work with. You hear a lot of these challenging stories that people face. And I think we've really been very lucky in that regard.
That is great to hear. Speaking of venture capitalists, how have they evolved? And I ask this because was there a message that is striking today as opposed to like 4 or 5 years ago? Have you seen change of scene from the investor point of view, or is there a point that started clicking where like, oh, this is a message that they want to hear? Or take it reverse, were the mistakes you were making early on?
Yeah, I would say a piece of it comes from the nature of the industry that we're in, which is with our flagship product, for example, it's very much a research-focused laboratory instrument or bio tool that tends to be a very, it's a difficult space for investors to understand. There's folks that have a lot of experience, like Karthik mentioned. We're very fortunate that the investors that we have understand the space very well, and they know both the customer side, our customer side, I should say, as well as other technology developers and system manufacturers that they've supported successfully. So they've seen both the development as well as the end customer aspect of it across their portfolios. But there's a lot of investors for whom that doesn't fit their profile, just because it tends to be a reasonably capital intensive segment of companies that need quite a bit of money. First of all, hardware is hard. I think that's a well-known adage at this point. But then you throw biology on top of it, where I think people understand some of the clinical applicability and clinical application spaces to the degree that, hey, this is a tool, not saying us as a company, but broadly speaking that, hey, if there are tools that are working towards medical device developments and so forth, that can be a very easy story and market to try to understand. But with research applications, I think that can be a step removed for many investors, especially folks that are outside of the traditional mold of the biopharma or research tool investors in the past. So absolutely that piece of it, it's an identification process. It's a trying to build a network strong enough that you get connected to the folks that are the decision makers or are the VCs in this space. That is certainly a piece of it. Another piece of it, of course, is it is a funnel at the end of the day. There's quite a few companies that raise initial round, pre-seed rounds or seed rounds of capital. And there are quite a bit fewer companies that end up raising Series A's and B's and so forth. And part of it is a numbers game. Part of it is complete luck and timing. And you happen to get a conversation with someone on the right day. And then there's the other piece of it, which is technology development. There was a lot of development that went on where the story that we were telling investors when we closed our last round was very different than the story we were telling 3 years before that or 4 years before that. It could be that we were looking at the same market, but where you are as a product, where you are in terms of maturity of even the traction or the types of data or folks that you've worked with, all of that can be very different. And those can be very major inflection points, I would say, throughout the process, where it's not necessarily that it's not some diamond in the rough that you're trying to find necessarily, even though there is a piece of that to it. But it's also that you have to find it at the right time and find it when you're in the right position to be able to present a compelling offering to the market.
Great points. Great points. I always wanted this more of a philosophical question for me, like who drives innovation? Is it the users or the investors? I was talking to some high-flying startup earlier this evening, and they said they wouldn't change anything because that's what the investors want them to use, a process. When you're taking startups like yours who are developing these technologies and tools, that would actually, say, reduce the failure rates of clinical trials, which is about 90%. It's a huge, big deal. Like, what needs to happen to change that process? for both innovators, it looks like a vicious cycle. Everybody's pointing a finger at the other person and saying like, oh, they want us to do it this way. That's why we do it. And somebody went ahead and said like 90% of the venture investments fail and 90% of clinical trials fail. So they're all in the same thing. Status quo is fine. What in your observation could help change this? This needs to change, man. This needs to change. Any thoughts on that, Karthik?
No, I don't think it's an easy question to answer, but I mean, I think just the ecosystem, and we even take it a step further. We think about the investors, we think about the folks within a company, and then even when you think about research and academic research, we think about really all of those. And I think one aspect of it is just having very clear exposure, communication of ideas, communication of technology. I think it's very easy to get super focused on one aspect of what you think matters. And I think for us, we've definitely seen a lot of the investors, they hone in on the market side. That may be what they really think about. On the innovator side, or a scientist may be just focused on the technology piece. They may not be thinking, oh, is this going to return investment if I get capital on this? So I think thinking about all the layers that are involved to make the whole thing successful, I think just being aware of that and also getting lines of communication with all the groups is important. I know that doesn't really answer your question, but
it's a tough one. I'm not expecting a clear answer from that. Maybe I'll go a step lower. During your conversation with potential customers, I'm sure you had to talk with different parts of a pharma company. And I'm sure you found a lot of people who are okay with status quo. And how did you have to change their minds? How did that convincing process happen for them to adopt a technology like yours? Take your best case scenario, you flip them. Any lessons learned from there that others could use?
Again, I would probably put it into, there's multiple camps of folks. Some are very set in their ways and are not willing to
saboteurs.
And you try to get out of those conversations as quickly as possible. But I would say the ones where you do see that kind of light bulb flip moment, I think, is where it's just trying to ask questions. The end point about all these conversations is not, hey, I'm here to sell you something. That's not the point of any of these engagements. It's really about trying to dig down into, what do you do for this process? Clearly, your company makes X product, X biologic. What do you do for this process internally right now? And then people are more than happy to talk about what they're doing right now, especially because they tend to like to complain about the things that they're doing or things that aren't working or things that aren't going right. And the more you kind of feed into that a little bit, I think you let them convince themselves really that maybe this isn't the right way to be doing this. Maybe I've never thought about it. Maybe I've never actually stopped and thought, hey, this is just written in some SOP somewhere and I've been doing it since I got here, or I've been telling my team to do it, but do we have to do it this way? And I think you have to let them convince themselves. Otherwise, at the end of the day, anyone trying to come in and saying, hey, I have all the answers and you're doing it wrong is not going to work in terms of convincing, especially scientific-minded people. I think it tends to be a hard sell in those situations.
Let's talk about something else related, but being in the Valley, there's always a comparison between all this software and speed and fastness of startups. What are the kinds of things that you had to face in terms of comparison? And how do you build a wall between yourself and all that noise there? Because it's easy. Like everybody talks about, investors are like, oh, there's a $5 billion company I can invest in. Why should I invest in you? How did you evolve in that and how did you become stronger?
I think there's...
Do you have a good answer for that? We're still waiting to figure that out.
Being in this area, there's certainly a ton of innovation, tons of very cool work going on. And I think we mentioned earlier, it can be a numbers game. It can be, how do you find people where what you're doing resonates for them? And that could be, part of it is a numbers game. I think part of it is also, you don't necessarily take a shot at every single possible conversation out there. That may not be the most efficient. And obviously, these things take time. They take a lot of effort, and you want to play the numbers as high as you can. But I think some of it is also understanding who is the right person to target and who might be open to the type of opportunity that you're presenting, and could they be a good partner for that? And I think refining that circle of candidates is one aspect of making it more efficient in the overall process. And you can see, have they done similar types of deals, or whether it's a similar type of technology value proposition, or in the broader space, have they had a success in recent years? And tapping into that success point and saying, hey, we could be the next XYZ, that you've had a good experience with this. Do you think this could be just as good or even bigger? And I think understanding the right person or the right partner to engage with, I think, is part of the work in itself. But at the end of the day, there's always going to be other opportunities that people could be looking at and they should be looking at. To your question, I think there's many different areas you can position your investment, and it's our job to sell it as well as we probably can, but also identify the people where it could be a good fit, I think.
I think that filtration is very, very important. And we mentioned about customers earlier on. How did that customer relation evolve from when you first presented that idea to when you want them to work with you? Like, did you have to do stuff for free in terms of proving your technology? How did that relation evolve over the past few years?
Sure. I'd say it totally depends based on the types of customers, based on the profile that they tend to fit and their need.
What was your dream customer and what was the worst? You don't have to name names.
I mean, dream customer would be folks that they would come up to our booth, for example, at a conference or heard a talk on a virtual conference or an in-person and reached out. I mean, obviously it's, as humans, we're inherently lazy. So if they do all the work for you, that's a dream customer, right? At the end of the day. But if they have clearly identified that, or if something in the presentation, especially if that is something that we've chosen to highlight, such as efficiency or affordability, or just fundamentally the ability to be doing things like single cell sorting on your bench, for example, if that's a concept that they didn't even know existed, if that wasn't something that had ever crossed their radar, but they think, I'm doing this every day. I actually have budget for this, and I am able to purchase something like this tomorrow or next week. And yeah, I'm going to go up to this company or I'm going to reach out to them. And that can be an incredibly easy sale to make at the end of the day. But I would say the ones that you learn the most from, obviously, are when that happens and it's a piece of the material or the piece of the presentation that you've made that wasn't something you're choosing to focus on. It was something that was almost mentioned in passing or mentioned as oh, this is another feature that we have, or this is just something that is on a slide, but someone comes out and kind of pinpoints that piece of it. They're like, oh my God, I could really use a system that does this and this. And I have never come across anything that does both of those together. And that may not have been something that we even really thought about as a way to position the platform or position the technology more broadly. And that's where you can get very interesting insights, potentially even sales, but really try to bring that knowledge of what are customers focused on, and is this indicative of something larger? Is this a bigger opportunity that we're not even thinking about yet?
Can you talk about that feature? One of those?
So I would say one of the pieces that was built out of customer feedback on the system is we're a sorter, we're an analyzer, and there's been many sorters and analyzers before, but we also, by nature of the work that we do and the methodology that we do it with the microfluidics, you can actually dispense, because it's a completely contamination-free workflow, you can actually use the system as a liquid dispenser. A very common use case for folks that are wanting to do gene editing workflows or CRISPR workflows is you need to isolate cells on an individual basis and then grow them out afterwards. It goes right into their incubator, they grow it out, and then they can use it for their downstream process, whether that's sequencing or whatever else they might be going through downstream of this. But there's multiple places that either they can't afford it or it's too much of a luxury to bring on board a top of the line liquid handling system or something like that in-house. So having a system that doesn't require their research associates time to pre and post fill well plates, for example, or be able to use that on a daily basis or do dosing studies across plate, because you have all the data of what was in the plate, what got sorted out. And so being able to do that process, I think, became something that was certainly something that we had never really advertised it. It was kind of we do this also just because of the nature of our technology and the way that we do the sorting and the dispensing of fluids out of the cartridge. But I'd say it's something that we are highlighting more and more now and been getting some pretty positive.
That's a great use case. Thank you for sharing that. Let us take a step back. You mentioned about the incubators that you went through, StartX and so on and on. Can you talk about one good point that helped you at each of these places?
I think one thing that's overlapping with, I would say, Berkeley Skydeck and Stanford StartX both gave us, I think that was very, very helpful, was just being in the environment, the ecosystem where there are other startups, other founders, other entrepreneurs around you. I think naturally, there's a lot of people, whether they're in your industry or not, that are going through similar challenges that you may have not faced yet, but you're going to face. And I think having that dynamic and sitting down and talking to folks like that it really allows you to get a perspective of, oh, am I going through something unique? Is this an atypical situation? Or, hey, we're having this problem. Have you guys gone through that before? I think just having the network around you to do that, and really, really innovative people, whether you're talking about Skydeck or Stardex, I think having that ecosystem, I think there's no real good replacement for being surrounded by people that are just looking to come up with the next biggest innovation and push it and make it commercializable and successful out there in the world. So I think we've learned a lot from others that are trying to build their own companies.
I would probably get murdered if I didn't mention that I met my fiance at Berkeley Skydeck. So certainly a very positive.
Congratulations on that. Yes. Before I go to my traditional closing questions, any plugins from the company side, or are there things that we didn't cover? When you talk about a certain point about a company, people get really excited about. Are there things that I missed?
It's really just a journey. I don't know if you missed anything in particular. I think our experience would be through the years, both of us would vouch for the fact that I think you have some of the lowest of lows, but you also have some of the highest of highs. And I think that's one of the things you're signing up for when you go through this journey. And I think it's very, very rewarding when you've been in those low points, when you do get to feel those high points. And I think that's probably an aspect that I think anyone that's going through this type of thing would likely agree with, where just by human nature, I think some deviations from the norm that's really what this experience is about, whether that's down or up. And I think if you have enough ups to outweigh the downs, it becomes a very, very worthwhile process. And I think for me in particular, going through some of those hard times, I mean, I've actually talked to Anand about this. It's as long as you feel like you're growing and you're learning through everything, it's tough to have any regrets as you go. Because I think the nature of this job is you're questioning all the time in the lows, like, should I really be doing this? What am I going through here? And we are fortunate enough to have families, whether that's parents, relatives, but also even our significant others that have been along for the ride. If not, they are driving the car. We would not be able to do this without that kind of support. And I think I certainly feel like we are fortunate to be in the position we are, because there are people that are okay knowing we spend more time with each other than we do with any of them. It's been a long ride, but I think if you can savor the positive moments along the ride and look forward to the opportunity ahead, I think that's what's kept us sane throughout all the low points.
Great observation. Anand, anything from you?
No, it's a hard one to top. Yeah. Echo everything that was just said.
Awesome. Well, let's go to the next question about company plugins, any job openings and any messages to investors. And close it with, what is that future going to look like that investors don't get or customers don't get 5-10 years from now? What is that beautiful future vision that you have? If you're very successful, how would it help the world?
Plugins, I'm happy to share a few. So we've talked a lot about the NX1 product that is actually going to be launching later this year. And again, designed to be the easiest to use cell sorter and single cell dispenser that's really meant for universal scale adoption by scientists that may have never touched flow cytometry or cell sorting instruments, all the way up to expert users or the biggest biotechs that may already have access to very high-end instruments, but are looking for decentralized solutions for all of their scientists, not just the ones that are exquisitely trained on some of these instruments. And yeah, we're hiring like crazy. So we've been expanding the team quite a bit already, but we're really growing across sales and marketing, manufacturing, quality, engineering, software, application scientists, technical support, anything you can think of. We're kind of expanding across the board. And we'd love to hear from any of your listeners or anyone in the audience that happens to know someone that is really passionate about making biology and making tools more accessible across the industry. That's really our dream as a company. That's our dream as founders of the company. Is the shining vision really is there's a lot of products out there that they certainly can push the limits of biology and what's possible and really what we're able to understand, but they tend to be what we consider kind of in the class of Lamborghinis. They're amazing products. They're beautiful to look at, and there's a lot of great engineering that goes into them, but most people are staring at them through a window. That's been a huge problem across biology, and that's not just in the West, by the way. That's kind of across the globe. And really, for us, the vision is not just with this product, and although we certainly hope that's the case with this product, but really as we grow as an organization and as we build pipeline cartridges and instruments and software offerings, the idea is really around the central theme of making tools more accessible or making biology more accessible broadly. So when there are new discoveries or when there's new innovation that's happening, a big piece of it should also be the trying to get it in the hands of any user. It shouldn't be restricted to just the top 1% or 0.1% of academic centers or clinical research centers. It should really be for any researcher that reads a paper or wants to see that, hey, I can do this work and I have tools that'll let me do this work without having it be a PhD thesis involved to try to figure this out. And I think that's something that we share. Our whole team certainly shares this idea of trying to develop tools, develop accessible tools that help along the continuum of healthcare, trying to make sure that even though we don't directly impact patient outcomes today, or we're reasonably far removed from it, that the tools that we're building are critical pieces of the workflow that may end up becoming new drugs or new solutions in and of themselves.
Absolutely. Thank you for that plugin. And these affordable tools are really, really helpful, I think, if you need better drugs. And good luck with all those things. Now, on to my traditional closing questions. Karthik, maybe you want to go first. Any lessons from mistakes to avoid while building Nodexus that people can take away from?
Oh, man. I think we literally make mistakes every day.
One or two big ones that you don't want others to commit.
One I would say is, I think, if I think about when we were starting this company, one of the mistakes I think I personally made was I was coming out of grad school and I was thinking, starting this company sounds really cool. Can't wait to grow it very quickly, have people where I can just kind of drive the decisions and they can run stuff in the lab. And I think it's very, very quick adaptation and learning that you really do have to be embracing the position, which can be, as a founder, especially early on in the company, you have to be that janitor, that engineer, that biologist, anything that's needed to be done, whether that's something you like doing or not, you have to be willing to take that on. And I think the sooner you can understand that and appreciate that, I think it's very, very beneficial. And I would say I learned that and appreciated that relatively quickly, but it was not my day zero thinking. So I think that's learning that I think I would say anyone should hopefully come in with that mindset at least.
Great point.
I will say you would certainly savor it more also when you're in that position and you do, if we're fortunate enough to keep growing and you can remove some of those tasks or those go to other people, you can savor that so much more, knowing that, hey, either I get to show someone the things that I learned, or, hey, this is something that I really didn't like doing, but I found someone and I hired someone that actually loves doing it every day. And awesome. That's a win-win for both.
Great points, both of you. And are there like one or two people that played a significant role in your success building Nodexus so far?
One or two is too few.
It can take more. That's fine. Yeah.
We mentioned our families, but Hanny is the other person. He's not on this call right now, but he's been with us, gone through the whole ride and same sacrifices. And now I think he's really doing a great job leading the engineering with more people involved in the company at this point. And I think we always appreciate that he's kind of stuck with us and really helped us develop this initial product and also some of the other stuff that we've been working on. Beyond our internal team, I think we have a number of advisors that have been very, very crucial. The folks at Skydeck, Stanford, some of the other program, QB3 that we've gone through. I mean, there was some early investors. If I had to pick one name, I'll probably say Dennis Chan. He's one of the very, very early angels that was instrumental in getting the company off the ground and really being on board with, but also guiding people that really had no idea what they were doing at that point and putting up with us through the ride and just being genuinely supportive. Sometimes it is helpful when you call somebody that could be disciplining you effectively for the headaches that have gone through, but is really just that voice of support and saying, hey, guys, we'll get through this type of thing. I think we could not have asked for a better initial group of people, and Dennis was definitely key in that, I would say.
Anand, anything from you?
I'd echo all of that. I would add some of our great new investors, Tess Cameron from RA Capital and Mike Polini from Section 32 and Bill Rhodes, who joined our board as part of this latest financing, but had been an advisor to the company. Prior to that, there's really just genuine, decent people. And Karthik mentioned it earlier, but we've been very fortunate to be helped along the way. And as he mentioned, kind of folks that will put up with us and will also be able to support us and allow us to really learn from them and pick their brains and really grow as founders. We had entered a new phase of the company that was drastically different than anything we had had in the years prior. And I think we couldn't have asked for better partners to be going down this road with.
Excellent. Anand and Karthik, thank you so much. This was so much fun. It's always good to talk to friends like you. I've seen you grow and wish you and the team lots of success. I would really hope to bring you back at the other end when you succeed and whatever you define that success as. But good luck with everything.
Thanks
Naresh.
Thank you for listening to this episode of the Lab to Startup podcast. You can find links to the resources and programs mentioned in these episodes, connect with Naresh, or subscribe to this show at labtostartup.com.